So I am currently a postdoc at the University of Edinburgh with Professor Siddharthan Chandran, but the talk that I'll be discussing today would mostly be around my PhD project, which dealt with modulation of bursting or network activity in Fragile X neurons and modulation by the astrocytes specifically. So all of this work was done in India, NCBS in STEM in Bangalore, and with a huge collaboration with, again, the University of Edinburgh with Professor Siddharthan and Professor Peter Kine's lab over at Edinburgh. So thank you for having me. Right. So just to give a brief introduction, what is Fragile X syndrome? It is caused by the hypermethylation of a trinucleotide expansion in the FMR1 gene, and it is the most common genetic cause of autism spectrum disorders and inherited mental retardation. So what happens in Fragile X is there is a loss of the Fragile X mental retardation protein, which is an mRNA translation repressor. Now, because it is an X-linked disorder, the incidence is higher in boys than in girls, and some of the symptoms include developmental delay, seizures, anxiety, sensory hypersensitivity, below average IQ, ADHD, amongst others. So Fragile X syndrome has been studied in great detail, and it has primarily been studied using animal models, and most of the work has focused around the glutamatergic synapse in the FMR1 knockout mouse neocortex. And what some of these works have told us is that in Fragile X, there's a decrease in long-term potentiation in the cortex. There's an increase in long-term depression, specifically mGlu-R-mediated long-term depression, and there's an increase in the number of spines. So what this has also led to is development of several drugs over the past years from data from these animal model studies. And these drugs have also undergone several clinical trials. But the clinical trials have been less than, the results of the clinical trials have not been as successful as anticipated. So what I wanted to do during my PhD was to explore the pathophysiology of FXS in a more human-based model, which is where the human pluripotent stem cell derived cortical neurons come into picture. So what are stem cells? Stem cells are cells that have the capability to differentiate into other cell types, and they can self-renew, which is very important. So we can get stem cells from the embryos, and they are called as embryonic stem cells. But one of the main points of the project was to look at what happens specifically in the patients, which is why we got stem cells from Fragile X patients. And these stem cells, we got adult cells from Fragile X patients, and these cells were reprogrammed to become stem cells. And this process is called induced pluripotent stem cell. Well, the cells are called induced pluripotent stem cells. And from these iPSCs, we generated neurons, astrocytes, and the lab in Edinburgh generates oligodendrocytes and microglia as well. So one of the great sort of potentials of iPSC cells is that not only can they be used to sort of understand the disease mechanism, which is what I use them for, but they can also offer important sort of platform to translate them, to use them as high throughput drug screening models, which is again what Siddharthan has been doing with ALS. So the first step of the project was to generate stem cells which will lack the FMRP protein. So here we have a cell line from a control individual, and here we have two Fragile X stem cell derived, Fragile X patient derived cell lines, And as you can see, the FMRP protein is absent in these two cell lines. In order to be absolutely sure that it is the lack of FMRP and not any other sort of environmental or genetic factors that could be giving us our phenotype, what we did was we made an isogenic embryonic stem cell line where using CRISPR-Cas, FMR1 was knocked out. And here as well you can see that because FMR1 was knocked out, there is no FMRP. And from these stem cell lines, we generated neurons and astrocytes, and the culture, the monolayer culture, had about 95% purity for both neurons and astrocytes. So one of the first questions that I asked was, do these stem cell derived neurons from Fragile X patients, do they have any differences in their physiological properties? And to do this, I co-cultured these human stem cell derived cortical neurons with mouse astrocytes, because one of the caveats of using stem cell derived neurons is that it takes a long time for the cell to mature, and I wanted to finish my PhD before 10 years. So I co-cultured them with mouse astrocytes. And after these co-cultures were 8-week-old in vitro, that is when I performed whole cell patch clamp onto the neurons. So all throughout the talk, what I'll be talking about are these spontaneous bursting events fired by the neurons. And the two parameters that we will mostly look at are the burst number and the duration of the bursting. So this is what the bursting profile of a control neuron looks like. And when I patched on from a Fragile X neuron, as you can see that the bursting pattern is completely different. So this was quantified here in both the control lines. We have a lesser burst number and a concomitant increase in the burst duration, whereas in the Fragile X cells, both in the iPSCs and the ES, we have an increase in the burst number and a decrease in the burst duration. So in order to go a step forward and actually sort of see what could be causing these differences, we looked at the persistent sodium channel. Now, persistent sodium channel has been implicated in bursting activity since, well, before I started my PhD definitely, since a long time. And one of the papers using a Fragile X knockout mouse model actually saw the actually sort of associated FMRP and persistent sodium channel, which is why it was an interesting candidate to look at. So how I recorded the currents was I used a slow depolarizing voltage ramp that went from -100 to -20. I record the baseline current at TTX, wait for 5 minutes, and then inject the same depolarizing protocol again, and then the subtraction of the two traces would give the persistent sodium current. So here we have the persistent sodium current from a control cell. As you can see, it's a robust invert current, whereas when I record from a Fragile X cell, there's a decrease in the persistent sodium current density, both in the iPS and in the ES derived cells. So with the mouse co-cultures, what we found was that there's a difference in the electrical activity between the control and the Fragile X, and this was mediated by a decrease in the persistent sodium current in the Fragile X neuron. But the main aim of the project was to always look at a completely human system. And so we wanted to ask, what is the potential, or do the human astrocytes even have a potential role in this network activity? So the next part of the experiment was to co-culture the HPSC-derived cortical astrocytes with the cortical neurons. And again, when these were 8-week-old in vitro, that is when I performed whole cell patch clamp. And here we have a control human neuron with a control human astrocyte. As you can see, it has the characteristic control bursting pattern shown by an increase in duration and a decrease in number. This was all well and good. And then we have the Fragile X neuron co-cultured with the Fragile X astrocyte, and again it has the similar Fragile X bursting phenotype, again given here by a lower burst duration and a higher burst number. So do astrocytes modulate the neuronal network activity? To answer this, we co-cultured a control neuron with Fragile X astrocyte and a Fragile X neuron with control astrocyte. So what happens when we co-culture a Fragile X neuron with a control neuron with Fragile X astrocyte is there is a flip in the bursting pattern, and the neuron now fires like the Fragile X, which is quantified here. Similarly, when we have a Fragile X neuron co-culture with a control astrocyte, the neuron now fires like the control. So the presence of human astrocyte is actually governing the bursting phenotype. So then the next obvious question was to ask if the persistent sodium channel could be modulated by these astrocytes. Again, I followed the same protocol. Here we have the persistent sodium currents from a control neuron with control astrocyte, which has a robust inward current, and a Fragile X neuron with Fragile X astrocyte, which shows the diminished persistent sodium current, as is shown in the quantification here as well. However, when we have a control neuron with Fragile X astrocyte, as is shown by the red trace here, there's a diminished persistent sodium current, and when we have a Fragile X neuron co-cultured with control astrocyte, there's a robust persistent sodium current. So the presence of astrocytes, not only does it modulate the bursting activity, it is also modulating the persistent sodium channel on the neurons. So how is the astrocyte doing it? Is it secreting something that could be having these effects on the neuronal network activity? To test this, what we next did was we collected media from the astrocytes. This is called as the astrocyte conditioned media. And then we added the media onto a neuronal monoculture. Now again, when these were 8-week-old in vitro, that is when I performed whole cell patch clamp to test the effect of the astrocytic secretome on the bursting activity. Similar to the previous slides, here we have a control neuron with cultured in presence of control ACM, and it was no surprise that it started to burst like the control. Similarly, when we have a Fragile X neuron cultured in presence of Fragile X ACM, it has the characteristic Fragile X bursting phenotype. However, when we have a control neuron now with Fragile X ACM, cultured in presence of Fragile X ACM, the control neuron now again bursts like the Fragile X. Whereas when we have a Fragile X neuron with the control ACM, the neuron bursts like the control. So there's a rescue of the bursting defect, and we see the neuron now bursting like the control just in presence of the secretome of astrocyte. So again, what I asked was if the secretome can modulate the bursting activity, can it also modulate the persistent sodium channel? So similarly, we have a control neuron cultured in presence of control ACM, Fragile X neuron cultured in presence of Fragile X ACM. It shows the characteristic persistent sodium currents that we had found in the control and Fragile X systems before. So here you have a robust persistent sodium current from the control, diminished persistent sodium current from the Fragile X, also seen in the quantification. But when we have a control neuron in presence of Fragile X ACM, there is diminished persistent sodium current, and when we have a Fragile X neuron in presence of control ACM, there's a robust persistent sodium current. So the presence of astrocyte and the astrocyte secretome can actually modulate the electrical activity of the neuron. So when we were doing this study, there was a paper that came out which showed that this molecule called S100 beta which was secreted by the astrocytes, actually modulate the rhythmic burst firing of neurons. So we wanted to see if it could be this potential molecule which was also involved in our study. And to check this, what I did was I recorded baseline bursting activity from the neurons, and I added S100 beta. And then I recorded the bursting activity again in presence of S100 beta. So here we have a control neuron with control astrocyte, the characteristic control bursting. And in presence of S100 beta, I do see some effect of S100 beta, but it's not enough to sort of revert the phenotype. And in terms of quantification, I don't see any significant difference between the burst duration or the burst number. And here we have a Fragile X neuron with Fragile X astrocyte with the characteristic Fragile X bursting pattern. To this, when I add S100 beta, as you can see, there's an increase in the burst duration and a decrease in the number from the same neuron, which is also shown here in the quantification of the burst duration and the burst number. Sorry, gave that away. Anyway, so yeah. So the same effect can be seen when S100 beta is applied to a control neuron with Fragile X astrocyte, which has the characteristic Fragile X bursting pattern. And when S100 beta was added to this neuron, there's a rescue in the phenotype. Whereas when S100 beta was added to a Fragile X neuron co-cultured with control astrocytes, I don't see any significant effect of the molecule. So the same question, if S100 beta was modulating the bursting phenotype, was it also modulating the persistent sodium currents? In the control, it wasn't, because after addition of S100 beta, I don't see any significant difference in the control. In the Fragile X neuron, which has a diminished persistent sodium current, when I add S100 beta, there's a significant increase potentiation of the persistent sodium current. Similarly, when I add S100 beta to a control neuron with Fragile X astrocytes, there's again an increase in the persistent sodium current, but I don't see any change when I add S100 beta to a Fragile X neuron co-cultured with control astrocyte. So just to be absolutely sure that it was the persistent sodium channel that was actually involved in this, we also perform simultaneous experiments with veratridine. And what we see that in a Fragile X neuron with Fragile X astrocyte, in presence of veratridine, there's a potentiation of the persistent sodium current, and which leads to a rescue of the phenotype here. Similarly, when veratridine was added to a control neuron co-cultured with Fragile X astrocyte, again there's potentiation of the current leading to rescue of the phenotype. So this was what I basically wanted to discuss today. And the major conclusions from this study that we drew was that the Fragile X cortical neurons have aberrant spiking activity, which is distinct from the controls. And in an all-human system where the astrocyte is also HPSC derived, the astrocyte genotype determines the neuronal phenotype. And just the presence of conditioned media from the astrocyte-only culture is sufficient to modulate the bursting pattern, both in control and the Fragile X neurons. And we think that it is the different levels of S100 beta in the ACM which determines the electrical activity in cortical neurons. And S100 beta secreted by, so to speak, healthy astrocytes can correct the firing defects in the Fragile X neurons by enhancing the persistent sodium current. So the major implications of this study, and forgive me for the schematic, art is not my strong point. There's a potential role of glia in Fragile X syndrome. And what this study basically provides is a framework to study further sort of neuron-glia interactions and see how that can modulate disease mechanisms. So I would like to really thank my PhD supervisors, Professor Shona Chatterjee and David Wiley, and my current postdoc supervisor, Siddharthan and Bhuvanesh, and also Peter Kine and Dr. Matt. So all of this was done over at NCBS INSTEM, which has a beautiful campus. And if you're ever in Bangalore, I would suggest visiting it once. And thank you so much for listening to me.
So we should have time for some questions. I'm going to ask a quick question first. The persistent sodium channel is often tied into NaV1.6.
Yeah.
Is there a NaV1.6 channel blocker you could interconvert?
So this came out after I finished my PhD.
Sure.
But there is a form of TTX that only blocks NaV1.6. It was also published by the same group that showed that S100 beta was modulating the effect, right? And they have actually shown that 1.6 is the isoform that is modulating the burst firing.
Okay. Bernard?
Yeah, thank you. It's very nice work. So I have 2 points here. Apparently, you have a mixture in your phenotype. You have a mixture of change in intrinsic properties. You don't add synaptic blocker in your preparation.
No, I don't add synaptic blockers.
So in terms of the intrinsic properties, you should see something if you record by patch clamp.
I don't. So
you don't see any difference in the rail-based current, in input resistance. I mean,
no, I don't.
But you have this persistent sodium current that is there.
That was sort of that is what stumped us as well. So I don't see any changes in the intrinsic properties, but it could also be. So when you're recording from culture-derived neurons, there is a lot of variability.
You have a mixture. Actually, these are differentiated cortical neurons. So it includes both inhibitory neuron and glutamatergic neuron.
No. This is just glutamatergic neurons.
Okay.
It's only excitatory cortical neurons. We don't have inhibitory neurons in the culture.
But probably depending on the on the density of plating, the pattern of firing is very depending on the density of your preparation.
So we make sure that the plating density is 35K all the time, but and I do see significant variability between batches of culture, but when I have the batches together, I don't see any difference between the control and the Fragile X.
Okay, thank you.
Okay, thanks. A question at the back.
Many thanks for the talk. The bursting behavior of the control neurons, it reminds me, at least face value-wise, some thalamic neural bursting pattern, which is basically low threshold spiking, where the T-type calcium channels are known to play a huge role. Have you had a chance to look at this?
I have looked at different ion channels, and what we have seen is that in addition to persistent sodium channel, there's the potassium dependent calcium channels, the BK channels, which are sort of defective in the Fragile X neurons, so they are low. And from my other experiments, I've also seen that KV7 channel is impacted. So in addition to persistent sodium channel, there are also other channels which not only determine the burst phenotype, but are also deficient in the Fragile X neurons. Why I have not showed that to you is because we couldn't sort of rescue any of the defects after adding a potentiator, and this was sort of like a clean story. So.
Thank you.
Thank you. Thank you for this talk. That was very clear. But my question is, it seems from what you've shown that every effect of the co-culture could be reproduced just with the conditional media, but I guess it's not that simple, right? I guess.
I mean, getting effective conditioned media itself is difficult, but what I think the lab has been working with conditioned media for years now. So we do have a protocol where we can extract sufficient conditioned media and add it onto the neurons.
Yeah, but the question is, you don't have any
It's
additional
not,
effect of the contact of astrocytes with
No,
neurons?
I
It's
don't.
just the
I've-
media, it's just secretion what makes the difference?
I mean, at least here, it seems to be one of the things is the media. Obviously, so what I have seen is that when we have astrocytes in the culture, the neurons are really happy. So obviously, there's
Yeah,
some contact
that's what I meant.
mediated mechanism. And as compared to when I add just the ACM, is just that for this phenotype, the ACM seems to be enough to not only give us the phenotype, but also to revert if we have the control versus the Fragile X.
Okay, thank you.
Okay, maybe one last question at the back.
Hi, thank you for your talk. Regarding your secretome, have you considered looking at the exosomes that are released, at least preventing extracellular vesicle release, or
Yeah,
looking at microRNA or the protein profile, or would this be something too tedious to identify?
I mean, that was one of the aims that we did want to sort of veer towards, but I have not done that in my work. But what I would assume is that if people are sort of continuing this work, that would be the direction to go to, because there was, one of my colleagues did an ELISA screen, and they found that the S100 beta, the protein levels of S100 beta in the astrocyte, in the Fragile X astrocytes are indeed low. So that would be something to consider.
Thank you.
Thank you.
Okay, so maybe in the interest of time, we're going to move on to the next talk. So I just want to thank Shreya for a great talk. Thanks again.